Spectral cytometry is awesome. It gives us the power of rainbows to understand single cells, but it can also be really confusing. My name is Peter Mage. I'm a principal engineer in the Advanced Technology Group at BD Biosciences, and today I want to talk to you. If you're feeling a little bit mixed up by spectral cytometry, all these crazy colors and terms and math, well, let's see if we can get ourselves unmixed. And we're going to do that today with finger paints and linear algebra. Before we do that, let's understand what do we mean when we talk about spectral cytometry. Fundamentally, what a flow cytometer does is measure biology by measuring light. We have proteins or other molecules that we want to learn about on our cells, but they're invisible. So we encode that molecular information with uniquely colored light through fluorochrome tagged antibodies. The amount of colored light we measure from each of our fluorochromes tells us something about the amount of each protein or biomolecule on our cells. To understand full spectrum cytometry, what we need to understand is how signals go from cells to light to data that we can interact with. So let's break that path apart. What happens is each of those colors or fluorochromes is going to emit light in proportion to their abundance on each cell, giving us some total new color that's representative of the sum of each fluorochrome's contribution. What a cytometer sees is some discrete sampling of that emitted light over the wavelength range that the cytometer measures. And then here's the important part. What we do is take that sampled total emission spectrum and we do some inference. Looking at the total spectrum we measured, we try to estimate what amount of each underlying color, the number of blue and green and orange molecules, would explain the signal that I measured. And that's what's going to give us our final estimate of biological abundances. Spectral cytometry changes two key steps in this signal transformation pathway compared to conventional cytometry. On the side of hardware, we simply have more detectors than fluorochromes, typically measuring across the entire visible wavelength spectrum with minimal gaps between those detectors. On the software side, we use some math called spectral unmixing instead of math called compensation, and I'll explain a little bit more about that in a minute. Okay, I said we would be trying to get less confusing, and that was admittedly a little complicated already. Let's take a little art break and see if we can loosen up our brains a little bit. I think it's a common fact that people love making new colors. That's one of the fundamental joys of painting and art is that we can create something new by taking existing colors and mixing them. So I know if I take blue and yellow paint and mix them up, I get green paint. But what's really fun is if I mix those paints in different proportions, say more blue and less yellow, I can get something like cyan or turquoise. In fact, this is a career opportunity. For those of you who are maybe growing disillusioned with science, I can guarantee you that spectral unmixing not only can, but will make you famous on TikTok, as shown by some of these accounts, which are able to look at a sample paint chip and almost automatically identify the exact constituent paints that need to go in to reproduce that. I include this because obviously every TikTok star's dream is to have their content used to explain scientific concepts. So I'm glad that we can help them with that. But this reveals something kind of fundamental about our understanding of color. Not only can we take colors and mix them and create a new color, if we see a color and we know which primary colors were available, our brains can start to guess or estimate how much of each of those primary colors went into that new color. So if I see green paint, I can probably be sure that if I only had blue and yellow paint, they were mixed in equal proportions. If I see a turquoise or cyan paint, I probably know that I used more blue and less yellow paint to achieve that. Well, hopefully the connection to cytometry is starting to become a little bit more clear. Just like painting allows us to create new colors by mixing primary colors in different amounts, cytometry allows us to create cell-specific colors or spectral signatures by combining different fluorochromes in different amounts. So for example, if I have some blue emitting Phe and some yellow emitting Pe, if I mix those in different quantities, one cell may have more blue and less yellow. One cell may have equal amounts of blue and yellow, giving us a new representative color for each phenotype represented by our cells. And just like our brains can estimate the underlying amount of each primary color that went into creating a new mixture, a cytometer can infer how much of each of those individual single fluorochromes was present in the final measured spectrum that I measured on a specific cell. We can take this even further to understand some phenomena that we commonly see in spectral cytometry. What about dyes that have very overlapping emission spectra, something like APC and Alexa 647? Well, let's think in terms of some paints. Let's say I had two shades of red, one darker like a Merlot, one a little bit brighter like a fire red or a ruby. And let's say that I mix those together to create a new color. If I was to take a poll and ask, okay, well, who thinks there's more Merlot and less fire red, or more fire red and less Merlot to create this new ruby color, we'd probably get a lot of disagreement about the exact amounts. In other words, it's hard to estimate how much of each individual color went into the mixture if those two colors are already really similar to each other. Now, of course, it gets hard to really connect this in a quantitative way to things like spillover spreading or other things that affect cytometry data. So maybe we can develop a little bit more rigorous of an understanding. And to do that, let's try and go back from thinking about how our eyes work to thinking about how a cytometer works. Just like a cytometer, our eyes contain photodetectors in a sense. Our rods and cones, primarily our cones for color vision, are sensitive to different parts of the spectrum to differing degrees. So we could think of our photoreceptor cells as some being more sensitive to blue, some more sensitive to green, and some more sensitive to red. And when we see a new color, our brains interpret that signal coming from those photoreceptor cells to see what we call a new color, like cyan. Well, in the same way, a cytometer is going to take a measurement from different parts of the spectrum with its different photodetectors. But instead of representing that measurement as a new color with a name, it's going to represent that measurement as a series of numbers, a vector, like I'm showing here. And although these seem boring to us, computers love these things. So essentially, the cytometer and the computer work together to represent a new color as a series of numbers. In other words, a cytometer sees color quantitatively. So let's try and tie this back to our cytometry problem. Essentially, if I know the primary colors or single stains that were used to stain my cells, I can infer their relative contributions to a total measured spectrum. Those relative contributions are in fact proportional to the relative abundances of biomolecules or proteins on my cells. And those are going to give me the biological expression patterns that I care about for understanding the phenotypes in my sample. Compensation that you may be familiar with and spectral unmixing that's a little newer, those are just the names for the specific math that we use to do that process of inference, to go from the color I measured and my knowledge of the underlying single colors to our estimate of the final amount of each single color that would best explain the signal that I saw. So what we're going to do now is try and understand, how does this actually work? And to do that, unfortunately, we do have to do some math. That's right, it's math time, but I'm going to try to make this a little bit more exciting. All right, so what we're going to look at is something called the linear mixture model. It's a mathematical model that describes how signals combine. So to do this, we're going to break out and look at how each of our individual colors behaves, and then we'll see how those signals are going to come together to give us our final measurement. What I can do is take each individual color spectrum by looking at its relative signal in each of my detector bands. I can then digitize that as represented by these little balls and come up with something like a representative single stain spectrum for each color. And then the final signal I get from an individual color is just going to be the product of that digitized per dye signal times the abundance of dyes. In this case, for blue, two of my dye molecule. That's going to give me my total contribution from the blue dye. I can repeat this for my green and my orange dyes, and then see that I'm getting a different contribution to the final measured signal from each of those colors. The total signal that I measure is just going to be the sum of each of those individual dyes' contributions. But of course, the cytometer doesn't know which photon came from which fluorochrome. It just knows the total number of fluorochromes that it measured in each of the detectors. So if we want to figure out the underlying amount of each of these colors, we're going to need to rearrange this problem to let us use some math on it. To do that, we're just going to rotate our representation and rewrite some things. So here what we're doing is representing each single color spectrum as a vector. And we're multiplying each of those vectors by the abundance of each of our fluorochromes. I can condense that representation further using something called matrix vector multiplication from linear algebra. And here, all I'm doing is taking those single color vectors, putting them into a single matrix. This is the spillover matrix that you may be familiar with from compensation, multiplying that by the vector of fluorochrome abundances in the middle, and finally getting my final measured signal on the right. Now, of course, mathematicians tend to like to condense this even further into a simple matrix vector representation, and I can give each of these matrices or vectors a variable name. In this case, I'm going to call my spillover matrix X. This is my reference single color spectral matrix. I'm going to call the abundance of fluorophore of each of the fluorophores F. This vector is going to represent each of those quantities. And finally, I'm going to have my mixed signal vector Y, which is going to represent what was measured on each detector. Okay, so we've shown how to describe the problem mathematically with the linear mixture model. How do we actually use that to learn what we want? Well, we know two things. We know the spillover matrix, or the single color spectrum of each fluorochrome, and we know that because we ran single stain controls. We also know what we measured on each detector because we just measured it on each detector. What we want to know is that value in the middle, the vector of fluorochrome abundances represented by F. This framing is called the forward problem. It kind of describes where the signal comes from if we know the sources. What we want to answer is what amount of each fluorochrome would explain the measured signal that I saw. And to do that, because we have the same number of equations and unknowns, we're just going to solve a system of equations, something that some of you may have done in high school math. To do that with a matrix representation, we're solving what's called the inverse problem. And in fact, this turns out to be something you do by taking the inverse of this matrix. That is actually what the compensation matrix is in conventional cytometry, just the inverse matrix of the spillover matrix. And we get our compensated data, F, by multiplying the compensation matrix by our vector of raw detector measurements, Y. Okay, so now we've rederived how compensation works, but how does unmixing work? Well, remember, I said that the essential difference between spectral and conventional flow is just that there are more detectors than fluorochromes. So I can represent a spectral situation by adding an additional detector to our little model here. Now I have 4 detectors and 3 fluorochromes, and you can see that all that really does is change the shape of some of the matrices and vectors in this representation. But in fact, the way that we describe the forward problem is identical. I multiply my spillover matrix by my fluorophore abundances, and that is where my raw detector measurements came from. I'm still trying to answer the same question. What amount of each fluorochrome would best explain the signal that I measured? But the difference now is because I have more equations than unknowns, there's not just one solution to this. There are technically an unlimited number of possible solutions. What we want to do is find the best solution, the solution that best fits the data we measured. This is, in fact, exactly what regression in the field of statistics does. We're finding the best fit of our observations in Y to the data described by X. And in fact, solving this regression problem through ordinary least squares, which is kind of the most straightforward approach, although there are other algorithms for unmixing, looks very similar to solving the system of equations that we did in the compensation case. Except instead of a square inverse matrix, we have a rectangular pseudo inverse matrix, but we still get our unmixed data in F by multiplying that pseudo inverse matrix by our vector of detector measurements Y. So hopefully you can see that even if all of the math doesn't totally shake out for you, the overall shape of the problem and the way we can describe it and the steps we go through to solve it is actually very similar between compensation and spectral unmixing. So to wrap things up, what we've seen is that spectral cytometry really does let us harness the power of rainbows to understand invisible biology. We can understand this at an intuitive level with finger paints and color mixing, and at a rigorous mathematical level with the linear mixture model and linear algebra. Hopefully, you can take all of these things together and, just like me, use this understanding to help become a little bit less mixed up by spectral flow. Thank you so much for your attention.